Hello, this is Gina Piscatelli with a lecture on the digestive system for Anatomy and Physiology 2. In this lecture, we'll look at the structure and function of just the large intestine. So first, we'll start with its major functions. I think its primary function is to absorb water. No nutrients are absorbed, really. And so the water absorption that takes place compacts any food residue that remains in the digestive tract. And of course, it stores this residue temporarily before it eliminates it, and we call the residue feces, so it eliminates feces. Now, I know I mentioned that no nutrients are absorbed from the large intestine, but there is absorption of some vitamins and some trace amounts of fatty acids. And these two or three products are from bacteria metabolizing either undigested material or making vitamins B and K. So the bacteria in our gut actually produces vitamins B and vitamin K, and those are absorbed into the circulatory system from the large intestine. No digestion takes place in the large intestine except for what the bacteria do. There are no enzymes in the large intestine. And in general, by the time the food residue reaches the large intestine, there's very little nutrients available. Some undigested carbohydrate, and the bacteria can use that to fuel the production of these vitamins and fatty acids. So feces contain just some water and then solids, including some enteric or intestinal bacteria, undigested fiber, fat, and once in a while some protein, usually not any carbohydrates except for the fiber. Also, feces contains mucus because, as we'll see, the intestinal wall has goblet cells, and so there's mucus production into the lumen. In addition, the feces contains, the breakdown products of red blood cells, which was transported to the digestive tract in the bile, and some sloughed epithelial cells, which probably come from all regions of the digestive tract, but primarily from the distal end, including this large intestine, because epithelial cells can be broken down through the digestive processes of the small intestine. So usually these sloughed epithelial cells are from the large intestine. So bacteria perform an important role in producing those vitamins, but there is a group of bacteria that we call enteric bacteria that consists of thousands of different types, and these make up the normal flora present in our gut. These bacteria enter from the small intestine, meaning from the digestive tract, or from the superior opening of the small intestine, of the digestive tract, or from the inferior opening, the anus. Regardless of where these enteric bacteria come from, they colonize the large intestine. And as I mentioned, they're very important for synthesizing vitamin K and B, and for breaking down indigestible carbohydrates like fiber. But what the bacteria do, since they're anaerobic, living in the gut, is they do fermentation. And fermentation of carbohydrates results in a lot of gas production, or sometimes some fatty acids, so you'll get hydrogen, nitrogen, methane, carbon dioxide, and some sulfuric gas. None of it smelling very good. The bacteria also perform important immune function for our body, keeping pathogenic bacterial numbers in check by competing for resources. So if we were to eat pathogenic bacteria, presumably they would not actually take root or hold in the intestine and be able to populate, because they would be outcompeted by these enteric bacteria, which do us no harm and actually help us with vitamin synthesis. So this helps to fine-tune our immune system and any inflammation that could happen in the gut. Now, there is a clinical condition related to one type of bacteria that is present in the intestine, large intestine of many people. And this clinical condition is known as antibiotic-associated diarrhea. So when a person takes antibiotics in large doses or for a long time, it can wipe out the competitive enteric bacteria that normally populates our gut. However, there is one type of bacteria that flourishes because it's antibiotic resistant. And this bacteria is Clostridium difficile. You might have heard of it as C. diff. And this is an anaerobic bacterium that many people carry in their intestine, but is kept in down to low numbers by the presence of other enteric bacteria. This Clostridium difficile can cause colitis, inflammation of the colon, and it eventually, through this process of inflammation, can lead to perforation of the large intestine wall, and then the bacteria in the intestine, as well as any other toxins, can escape into the bloodstream, and you can get sepsis, so widespread infection. So it is a problem, and there's like 14,000 deaths, I think, from this diarrhea, because you lose all the water and you lose nutrients, and it's kind of a wasting disease. But there are new treatments, including fecal transplants. And so you take the fecal matter from one person that contains healthy enteric bacteria and transplant it into the gut of a recipient, and that suppresses the colony growth of Clostridium difficile. I don't think I really want to have a fecal transplant if I can avoid it. Okay, let's look at the gross anatomy of the large intestine now. It runs from the ileocecal valve all the way to the anus. So the ileocecal valve is where the small intestine passes contents into the large intestine, and the anus, of course, is the opening to the external environment inferiorly. And there's 4 regions. The cecum, which we've seen in the cat, and the appendix. Humans have an appendix coming off of the cecum. And then the rest of the intestine could be referred to as the colon. We also can just call it the large intestine. And there's an ascending portion, often called the ascending colon, and then a transverse portion called the transverse colon, and a descending portion called the descending colon. And then there's this curved region, and that's called the sigmoid colon, which leads to the rectum, and then finally the anal canal, where the opening exists, and we usually refer to the opening as the anus. Now let's look at some superficial features on the exterior of the large intestine. Notice how there's these bulges these big sac-like bulges. Those are called haustra, pocket-like sacs, and they form due to the presence of some longitudinal smooth muscle strips called tinea coli. And I'm going to draw on top of these smooth muscle strips, tinea coli. There are 3 bands of smooth muscle that run in the middle of the diameter of the small intestine. So I'm kind of drawing on top of it. And there's a certain muscular tone of this kind of contraction. And that creates these pocket-like sacs for the kind of peripheral wall of the small intestine, which isn't contracting. Another superficial feature of the large intestine are these appendages that contain fat. They're pouches of the visceral peritoneum that have fat deposits. I really don't know the function of them, but they're called epiploic appendages. Okay, from gross anatomy now let's look at the histology of the large intestine and what makes up the wall. So the innermost layer of the wall of the large intestine is the mucosa, just like everywhere. And most of it, from the cecum all the way down to maybe the sigmoid colon, is lined with simple columnar epithelium with lots of goblet cells. And these goblet cells are found in infoldings called crypts. Crypts of Lieberkühn, we don't have to know that, just crypts. And so there's a lot of goblet cells creating heaps of mucus, which of course is just going to help propel the feces down the length of the intestine, large intestine. Below the mucosa, oh, I forgot to mention, sorry. In the anus, we have stratified squamous epithelium instead of simple columnar epithelium. Now in the submucosa, there are blood vessels running in connective tissue. And when we get to the anus, there are some veins that we're going to look at called hemorrhoidal veins. And all of the veins that are in the hemorrhoid or anal region, the hemorrhoidal veins, don't have valves. And so they're prone to venous pooling and cause kind of distension of the vein. If this happens to a large extent, we get a condition called hemorrhoids, which I'll introduce you to in a little bit. And that's when the veins are distended so much and dilated so much and they become inflamed and kind of itchy and painful. So we'll look at that a little bit more. But there's vessels in the submucosa. And then the muscularis externa has 2 layers of smooth muscle, and then the tinea coli on top of that, but that's not really the wall. The tinea coli is very external to the wall of the large intestine. Okay, let's compare the histology that we've looked at in the small intestine with what we just talked about concerning the large intestine. So the small intestine, of course, has this serosa on the outside, and so does the large intestine. And then there's 2 layers of smooth muscle shown in pink. One layer runs circularly and one runs longitudinally, and that's true in both the small intestine and the large intestine. And within the layers of smooth muscle is part of the enteric nervous system, a network of neurons called a plexus. And since it's in the muscle, we call it a myenteric plexus. And that's true in both the large intestine and the small intestine. Then we get to the submucosa. And in the submucosa of the large intestine, really the only thing that I mentioned were veins. But there is some mucosa-associated lymphatic tissue. It's very diffuse. But if you look at the small intestine submucosa, you'll see that the lymphatic tissue is more nodular. And we looked at Peyer's patches, aggregates of lymphatic cells that are present in the submucosa. That's different than the large intestine. The large intestine, it's more diffuse. In addition, the small intestine has Brunner's glands, which make mucus, and that extra mucus that's produced is going to help neutralize the acidic chyme that enters the small intestine. We don't see any Brunner's glands in the large intestine, but we do have mucus production, and that's because of the presence of these crypts having goblet cells in the large intestine, the mucosa of the large intestine. But the large intestine doesn't have an increased surface area for any kind of absorption because that's not its primary role. So you won't see any villi. Whereas in the small intestine, the mucosa is adapted both with crypts and villi containing microvilli. And the villi and microvilli help for absorption, and the crypts actually produce some hormones as well as have immune cells, Paneth cells, and produce mucus. So we can see that the small intestine is really adapted for absorption, whereas the large intestine is really adapted for just propelling, not absorption. Okay, if we look at the rectum in particular, you'll notice that the mucosa and submucosa have these inward folds. And I kind of pointed them out with arrows there. And these folds slow the passage of feces. So entering the rectum is the feces, and it can kind of get caught up here on the edge of this, we call it a valve. But because the valve doesn't go all the way across and actually close like a sphincter, it doesn't do that. It allows the passage of gas or flatus. And so that's how you're able to pass gas without passing feces at the same time. Okay, I mentioned hemorrhoids before, so I'd like to look at a little bit more detail at the anal canal and the sphincters associated with it. So here we are in the rectum with the presence of the rectal valve, and we come down to the anal canal. And at the beginning of the anal canal, you'll notice that in the submucosa are these hemorrhoidal veins. Okay, and that, of course, is carrying deoxygenated blood eventually back to the heart. But if you look at the muscularis externa, it's actually adapted. There's these two layers, the internal layer and the external layer form sphincter muscles surrounding the entire opening, the anus of the large intestine. And so the internal sphincter is composed of smooth muscle. But down here at the anus, instead of having smooth muscle like you would see for the rest of the digestive tract, you have skeletal muscle. So what that tells you is that the smooth muscle cannot be consciously controlled. That sphincter cannot be controlled. But the external sphincter, composed of skeletal muscle, can be voluntarily controlled. So before we look at the control of those muscles, let's look at the condition hemorrhoids, which involves these hemorrhoidal veins located in the submucosa. With excessive sitting, standing, pressure during pregnancy, these hemorrhoidal veins are prone to venous pooling, meaning the blood in the veins is not returning, because remember there's no valves in these veins, and so the venous pooling causes the distension of these hemorrhoid veins. And sometimes the hemorrhoid veins or the hemorrhoids occur kind of internally inside the anal canal, and sometimes out at near the opening where the anus is. So there's two different locations that they can occur, but the cause is the same, and it's due to venous pooling because these veins do not have valves. Okay, one thing I've neglected to mention is the role of the appendix in our digestive system. And it turns out the appendix has lots of lymphoid tissue, these kind of nodular tissues. Even though I said the large intestine doesn't really have it, here in the appendix we see some nodular lymphatic tissue. And this is important because also located in the appendix is colonies of all the enteric bacterial types that we have. So it's a storage site for enteric bacteria. And if the enteric bacteria is present next to the lymphatic cells, you can see that our immune system would not exactly become immune, but the bacteria have developed in such a way that our immune system doesn't react to them as much. And so this bacteria that's stored in the appendix can recolonize the gut when needed, like after a case of diarrhea or something like that. Because the bacteria are motile, they can move, they can leave the appendix, travel to the cecum, and travel throughout the intestine. Speaking of movement, let's look at how the muscle in the large intestine works. So there's two types of motility or muscular contraction in the large intestine. One type is called Haustral contractions and another type is called mass peristalsis. The haustral contractions involve the haustra, the sac, the pocket-like sacs that we looked at before. And so it's kind of like segmentation that we saw in the small intestine. Different haustra will contract in different locations of the large intestine periodically, and it generally occurs in the ascending and transverse colon. So it kind of just presses on the contents and moves it a little bit and mixes it up, which allows some water reabsorption. And then the second type, the mass peristalsis, is what we sometimes refer to as a mass movement. These are slow, powerful waves, like we saw in the small intestine as well, and can move the feces a much greater distance. So haustral contractions occur when a haustrum has been distended by the presence of feces. So the presence of feces distends the haustrum, and right away local stimulation causes the muscle to contract. We consider that to be a local control. Largely the enteric nervous system, but also kind of just a function of smooth muscle. When smooth muscle is stretched, it contracts. And this passes food residue a bit to either one or two distal haustra and occurs every 30 minutes after eating. But mass movements can move the residue for several centimeters with each wave of contraction. And it occurs after eating 1 to 3 times a day. And it eventually is responsible for propelling all the feces remaining from a meal to the rectum. Now, this mass movement process, this wave-like contraction, is again triggered by filling of the stomach, which we saw that was true in the small intestine as well. So as soon as food enters the stomach, it kind of puts the motility of the small intestine and the large intestine into action. Because in this case we're talking about the large intestine, it's called the gastrocolic reflex. So the stomach being filled causes a reflex in the colon. Okay, the last topic that we have has to do with eliminating feces. And the term for eliminating feces is defecation. And there is an involuntary process by which feces are eliminated, and then a more voluntary process. So I'm going to show you the involuntary process first. And since it's involuntary, we refer to it as a reflex, meaning there's going to be a stimulus, and then a sensory signal, and then a motor signal, and finally an action. This involves just neurons of the spinal cord. So you have stretch of the rectum, and there's a neuron that senses that in the wall of the rectum usually, or the anal canal. And this neuron's a sensory neuron located in the pelvic nerve, and it sends the signal to the spinal cord. Once it reaches the spinal cord, that neuron synapses on a motor neuron, which is a parasympathetic neuron. And the parasympathetic neuron travels to the sigmoid colon, the rectum, and the internal anal sphincter. So what are the actions? So the parasympathetic nervous system tells the sigmoid colon to intensify its mass movements, and the rectum to contract, and then it relaxes the internal anal sphincter. So what this means is that as soon as the rectum is stretched, so you have stretch of the rectum, it causes the sigmoid colon to contract, the rectum to contract, but the internal anal sphincter to relax. So presumably, as soon as something enters the rectum, it's going to be ejected. We know we can override this, but this is an involuntary reflex, and I'll just show you the network of neurons a little bit. So here's stretch receptors in the wall of the rectum connected to a sensory neuron that travels to the spinal cord, and it synapses on a motor neuron that travels to the sigmoid colon, it travels to the rectum, and it synapses on another one which travels to the internal anal sphincter. And so the action on the sigmoid colon is excitatory. The action on the rectum is excitatory, but the action on the internal anal sphincter is inhibitory, so that it relaxes. Now you know we can override this and wait to expel or eliminate feces until a more convenient moment. So we do this generally by keeping the external anal sphincter contracted most of the time. So it's constantly contracted until we consciously relax it. It is going to involve some autonomic neurons, but because the skeletal, and that's going to be mainly sensory type stuff, and inhibiting some motor neurons of the autonomic nervous system, But we're just going to focus on what happens to the external anal sphincter, because as you might recall, it's made up of skeletal muscle. And so that means it's innervated by somatic neurons, somatic motor neurons. And those somatic motor neurons are connected to a neural pathway that originates in the cerebrum, our site of conscious control. And so the cerebrum sends a signal that eventually gets to these somatic motor neurons, and they will inhibit contraction. The motor neurons will actually be inhibited. So when the cerebrum inhibits the somatic motor neurons, then you'll get the external anal sphincter relaxing. Now there is a point where this voluntary control isn't effective, but it would be in a not exactly a disease condition, but extensive pressure in the rectum will trigger relaxation of this external anal sphincter, regardless of our wishes to voluntarily control it. But let's look at the neural pathway of the voluntary control of defecation. So this involves the motor neuron traveling down the spinal cord. It originated, or at least the pathway originated, up in the cerebrum. And so this somatic motor nerve fiber has to be inhibited here. And so once the neuron is inhibited, it's not going to be releasing any neurotransmitter, and so the muscle will relax, the sphincter will relax, and that allows materials to pass through. But most of the time, unless the cerebrum inhibits this motor neuron, this somatic motor neuron fiber, most of the time it is releasing a neurotransmitter that is excitatory to this muscle. And so that helps to keep the sphincter closed. It's not until we inhibit that pathway that the muscle's relaxed. Okay, that's all for the large intestine. Thank you very much.